Welcome to this free webinar on OmicsLogic Data Science. In today's session, our team will be sharing information, examples, and an overview of OmicsLogic training resources designed for anyone to learn data science for biomedical data, as seen in application to projects sourced from current topics in biomedical research. My name is Deepsha Biswas, and I'm the OmicsLogic Community Manager responsible for daily interaction with thousands of users that are part of this bioinformatics community managed by Pine Biotech. Let me start by introducing Pine Biotech, the company behind OmicsLogic, and share with you details about the upcoming OmicsLogic programs and explain how the training resources we will be discussing and using through this entire training. OmicsLogic program for bioinformatics training was developed in collaboration between Pine Biotech and the Tauber Bioinformatics Research Center. What we will discuss today and throughout this program is a result of hard work by an amazing team led by company co-founders Dr. Alfred Tauber, who comes from a medical background and has extensive expertise in oncology, immunology, and biochemistry of inflammation, and has been recognized internationally for his generous support of many research projects and initiatives that support data science training and applications in medicine. Dr. Leonid Brodsky is the director of the Tauber Bioinformatics Research Center at University of Haifa and is an expert in bioinformatics and biostatistics. He has numerous publications in mathematics and bioinformatics, and over the course of his career has developed multiple novel algorithmic approaches to biomedical data analysis. The rest of the team includes experts in bioinformatics, computer science, and some of our team will be presenting today as well. Over the course of the past several years, our team has been collaborating with a number of academic institutions around the world that have participated in content evaluation, curriculum review, and project design. As a result, the OmicsLogic programs have been thoroughly evaluated and are known to lead to excellent outcomes. Importantly, the programs are designed to support students with minimal bioinformatics background, including biology and biomedical study majors, as well as faculty interested in bioinformatics, data science, and biomedical research. The goal of the OmicsLogic programs is to provide training in bioinformatics, enabling independent research guided by mentors and peer example, developing a growing community with shared passion for data-driven research and appreciation of citizen science, enabling students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life and data sciences. The program is designed to offer an introduction to statistical analysis and machine learning, and a broad overview of technologies and project examples to learn about and develop practical data science skills using user-friendly cloud technologies as well as R and Python coding. After completing this program, you will gain experience with methods for analysis of big data and data wrangling, essential skills for bioinformaticians, as well as those interested in other types of non-biology related research using data analysis. The program is designed for students starting from the undergraduate level to faculty and researchers from both biology and clinician backgrounds can participate to learn more about data science in biomedical research. In the upcoming OmicsLogic program, you will have the opportunity to interact and collaborate with an international team of experts specialized in the application of data science to biological data sets. Let me introduce you to the main speaker of today's session, We have Dr. Harpreet Kaur, who is a certified OmicsLogic trainer and an expert bioinformatician, and will guide the hands-on sessions and assist participants with projects and analysis during the program. Her area of specialization is cancer genomics and machine learning. Dr. Mohit Mazumdar is our OmicsLogic project mentor and an expert in big data computational analysis. Dr. Mazumdar has a PhD in computational biology and over 10 years of experience working with industry and academia. Ilya Brodsky is the co-founder and CEO of Pine Biotech. In his current role, he has extensive experience working together with industry and academic partners that are applying multi-omics integration and bioinformatics to a variety of biotechnology and clinical applications. I will pass it on to Ilya to talk about the increasing significance of data science in biomedical research and introduce you to the type of challenges we will be addressing in this upcoming program. Over to you, Ilya.
Thank you very much, Pizza. My name is Ilya Brodsky, as was mentioned, and I'm one of the co-founders in Pine Biotech, and I'm happy to share with you today a little bit of a perspective about big data in life sciences, and also talk about how this program is going to help you understand the role of data science and the significance of skills related to data science, as well as the application to different types of projects. So as you know, over the past 20 to 30 years, there has been a phenomenal growth in the adoption of high throughput technologies like next generation sequencing in research, biotechnology, and even clinical practice. These technologies produce an enormous growth of publicly available omics data, and we can leverage that data to understand how we can analyze and study biology at a completely new level. However, this trend has also produced a big data problem, and that precludes many biologists and clinicians from gleaning knowledge from these data regarding the nature of malignant processes and the relationship between clinical phenotypes and their molecular drivers. But what we know is that the same technology, like next-generation sequencing, can be used to explore the roles, relationships, and actions of the various types of molecules that make up the cells of an organism. Different types of information can be extracted from these omics data types. And the high throughput sequencing technology is just one of the technologies that allows us to study how these molecular processes happen at tissue, organism, or even single cell levels. Most of these data types are generated randomly at high speeds and producing large unstructured data sets. To process and structure them, they require complex computational data processing, a procedure that will convert the unstructured data to structured data, which can be visualized and analyzed statistically. Commonly, these types of data sets are used to study a variety of factors, including DNA variation, gene expression variation, protein abundance levels, and even composition properties of microbial communities. All of these are very different types of omics data. However, we will learn to explore and mine them for patterns using some standard tools that come from the field of data science or computational biology. For example, we'll look at how gene expression patterns can be associated with clinical phenotypes and help identify what's called molecular biomarkers that are used for diagnostics and therapy selection. We will also talk about how new discoveries based on analysis of such data include a better understanding of complex diseases at their subcellular level, as well as allow for target identification for novel drug design and repurposing. We will take a variety of research examples and learn about the logic behind each type of omics data that is used to study cancer, tissue biology, and the microbiome. We will also look at the full overview of data science and biomedical research to understand how different stages of analysis work together to help define the hypothesis or the question that we're asking, as well as prepare the data for meaningful application of machine learning to practical challenges. We will also have the chance to see how different data mining and machine learning or classification methods are used to study patterns, understand grouping between data objects by feature similarity, and how prediction can be achieved by training different types of machine learning and evaluating the model accuracy to understand how we can rely on such predictions. We will also speak about making machine learning models more transparent and easily explainable when we present the results of our analysis to collaborators that might come from a clinical or biological background. Now, let me pass it on to continue speaking about one such project example led by Dr. Harpreet Kaur, who is going to be the OmicsLogic trainer, so she can introduce herself and talk about her research experience. Over to you, Harpreet.
Thank you, Ilya. So just let me share my screen. So about me, my name is Harpreet Kaur and I have done my PhD in bioinformatics from CSIR Institute of Microbial Technology and GNU New Delhi. And I have done my master's in molecular biology and biochemistry from GNDU Amritsar, India. And I have done my graduation in biotechnology from Punjabi University, India. So about my research background, I have experience in the bioinformatics, cancer genomics, machine learning, biomarker discovery, and then resource development on different aspects of human health. And I have most of my research work is framed in the 14 research publications with having more than 200 Google Scholar citations. And for my different research projects, I have got ESL Jung Investigator Award and INC Jung Researcher Award. And I have received my doctoral fellowship from the CSIR, that is Council of Scientific and Industrial Research. Let me give you an illustration how we can proceed for research problems in data science with my own research experience. I will give you examples from my PhD research process. So my PhD is focused on computer-aided identification of genetic biomarker for the diagnosis and the prognosis of the liver cancer. So let me tell you what inspired me to take liver cancer as my doctoral research problem. Cancer is considered as one of the major concerns worldwide, as according to the Globocon report 2018, there are nearly 18 million people that are diagnosed with cancer every year, and out of which 9.6 million people are dying because of the cancer per year. So one of the 6 that is only due to the cancer. Among the cancers, particularly the liver cancer, it is considered as 6th major cancer in terms of the number of the new incidences that taking place every year. And in terms of the mortality rate, it is considered as the 4th major cancer that contribute nearly more than 8% of the mortality rate that is only due to the cancer. So to proceed a research problem, first of all, we search the literature and we have gone through what are the major challenges and what are the major problems that are associated with the liver cancer. So by extensive literature research, we found that there is 3 or the 4 major issues that are associated for the liver cancer with the liver cancer that eventually results in the low poor prognosis. In literature, we found that there is a very poor survival, 5-year survival rate for the liver cancer. That is nearly 18%. So, what were the factors that affecting its high mortality rate? So once we observe that, there is the first problem is that it's inaccurate cancer diagnosis or cancer prediction. So we have observed that as hepatocellular carcinoma, that is HCC, it is one of the major uh, form of the liver cancer, where cancer is derived from the hepatocyte. It nearly contributes 75% of the cases to the liver cancer. The second major primary liver cancer, that is cholangiocarcinoma, which derived from the cell lining the bile duct. And literature indicated that there is a lot of the high mortality rate is due to its inaccurate diagnosis. So we understand that there is need to identify the methods or the biomarkers that can predict cancer with more accuracy. Then we have observed that in the literature, if cancer is diagnosed at early stage, then there are a large number of the treatment options are available. So they become limited if cancer detected at the late stage or or you will detect a cancer at advanced stage. So the next problem that we have observed that there is need to identify a genomics biomarker that can predict cancer at early stage as possible. The third factor that contributes to the high mortality rate of the liver cancer, that is there is tumor reoccurrence taking place. So we have understand that we need to identify genomics or omics biomarkers that can identify the patients that are at high risk and the patients that are low risk. So accordingly, treatment options are available for every kind of therapeutic treatment for the particular class of the patient. So with this background, we have we have tried to address these problems by asking 4 major research questions. First, we want to identify transcriptomics biomarker that can classify HCC from the normal sample. So as I have already told you, HCC is one of the major primary liver cancer. The second question we have tried to address that we should able to identify transcriptomic signatures that can predict cholangiocarcinoma in comparison to the normal and the HCC samples. Then our next research problem is focused on the stage classification, where we want to identify multi-omics biomarkers that include transcriptomics as well as the methylation based signature that can predict liver cancer at the early stage. And in the last problem, we have tried to address to identify multi-omics biomarkers that include RNA, miRNA, and the methylation that can stratify high risk and the low risk reoccurrence HCC patient group. So let me demonstrate one by one some of these results. So first, in the first question, we have tried to identify transcriptomics biomarkers that can predict HCC from the multiple platforms. So for this, we have collected 32 datasets from the public repositories, that is GEO and the GDC data portal. So for all these 32 datasets are processed using different profiling techniques, that is Illumina, Agilent, Affymetrix, and high throughput. Further, we have divided these datasets into 3 categories. One for the training dataset, the second for the differential gene expression analysis, third for the external validation cohort. So to reduce the genes from large scale, as there are 20,000 genes are present in any kind of transcriptomics data. So to filter out the important genes, we have performed differential gene expression analysis on 27 datasets, which have nearly 1200 HCC samples and nearly 950 HCC non-tumor normal samples. And first of all, we have performed differential gene expression analysis on each of these datasets using Wilcoxon and Welch t-test. So we have obtained 26 genes that found to be significant differentially expressed in all these 27 data sets. So then to develop a prediction model or to confirm the prediction power of these genes, we have kept a data set training where we have nearly 250 HCC samples and 250 normal samples. So then we have on the training data set, out of these 26 genes, we have applied various feature selection approaches, including recursive feature reduction approach, where one by one gene is eliminated, whose deletion is not affecting the performance of a particular prediction model. So eventually we have obtained the 3 genes. So based on the 3 genes that include FCN3, CLAC1B, PRC1. So based on these 3 genes, we have developed machine learning prediction model using various classifiers like SVC, RBF, random forest, extra tree, LDA, etc. on the training data set. Then the most important step, you must able to validate your prediction model. So you must able to evaluate the performance of your biomarkers on external validation cohorts. So for this, we have 4 external validation cohorts where we have nearly 850 HCC samples and nearly 500 normal samples. So performance of these models based on these 3 gene-based signature is evaluated on all these 4 external validation cohorts. So we have observed a prediction model based on these 3 genes able to discriminate HCC and the normal samples with an accuracy nearly 97% and sensitivity and specificity is more than 90% in each of the data sets in its training as well as external validation cohorts. So after assessing its diagnostic potential, then we want to assess its prognostic potential. We want to understand whether these genes able to predict the prognosis of the patients. That is, whether it can predict the patient at have higher survival rate or patient at have lower survival rate. For this, we have taken 2 cohorts. One is from the TCGA, that is the Cancer Genome Atlas for the liver cancer. The other is GSC 14520 cohort from that we have obtained from the GEO. And in the TCGA, we have nearly 350 samples and the GEO cohort, we have nearly 220 samples. So then we have performed univariate survival analysis to assess its prognostic potential. Here we have considered 2 types of survival data, that is overall survival and disease-free survival. This disease-free survival is also called as reoccurrence-free survival. So eventually we have confirmed that all these 3 genes able to significantly stratify high and the low risk of survival risk groups of HCC patients with an HR value, that is hazard ratio, more than 1.5 and p value less than 0.05. Eventually, all these prediction models were integrated in form of a web server that is called HCC Thread. So this paper is published in the January 2020 in the Frontier in Genetics. And this is the web server where we have integrated all these prediction model to predict the HCC sample, that is called HCC Thread. In the second problem, we have focused on the second major liver cancer type, that is cholangiocarcinoma. Cholangiocarcinoma, as I have already told you, is the cancer that derives from the cells lining the bile duct. So here we have focused on identifying the transcriptomic specific signature that can classify CCA from HCC as well as the normal sample. For this, we have obtained nearly 9 data sets from the GEO. First of all, we have pre-processed these data sets, and then based on some of the data set, next we have performed differential gene expression analysis. In the differential gene expression analysis, we have identified nearly 8,800 genes that found to be significant differentially expressed in CCA and the normal samples. And we have identified there are 229 genes that found to be significant differentially expressed in CCA and the HCC sample. Then we have compared all these genes and we have found that there are 202 genes that were found to be significant differentially expressed in the both the classes, that is CCA versus normal as well as CCA versus HCC. Then to further reduce the number of the features from this signature set, as you cannot, it is practically difficult to design a biomarker with such a large set of genes. So we have applied various feature selection and feature reduction approaches, including simple threshold, SVCL1, WECA, etc. Then based on the selected set of features, we have applied various machine learning algorithms. And here we have applied different machine learning algorithms to develop a machine learning prediction models on a training data set like extra tree, logistic regression, random forest, KNN, rich classifier, perceptron, etc. Then performance of these multiple genes is evaluated on validation, external validation cohorts. And we have here we have observed that we found the 3 gene set, that is LAMP2, ITZA, and C2, that can classify CCA samples from the normal and the HCC sample with an accuracy more than 90%.
Thank you, Harpreet. Thank you very much for sharing your background. Maybe you could also introduce Mohit when you get a chance. Thank you.
So now let me introduce Dr. Mohit, who is our mentor at Pine Biotech.
Thank you, Dr. Harpreet, for this wonderful presentation. So let me share my screen. Okay. So here we go. So my name is Mohit Mazumdar. I am one of the global representatives of Pine Biotech. And my PhD was focused on identification and prediction of binding affinity towards calcium for the protein. As a result, I had to master data mining and become proficient at predictive modeling. Our team ended up using combination of amino acid features to train multiple machine learning models using a nonlinear kernel implemented in support vector machine. That was really fascinating. In my current role, however, I am collaborating and working with academia and industry to provide support for bioinformatics education and research. I have 12 years of experience working and collaborating with pharma, healthcare industry to solve high priority scientific questions related to biomedical data analysis and also bioinformatics capacity building. So as Ilya mentioned, there has been a remarkable shift over the last decade in the cost of generating these molecular measurements. Most notably, if you think about DNA sequencing, but also I think the other omics technologies like transcriptomics, proteomics, and metabolomics. So most impacted by this change is the area of, I guess, genomic medicine, where it is now possible to move from generating reference on population level data to producing data from individuals, right? Concurrently, there has been advances in development of these new algorithms and tools to integrate and interpret this kind of data. So bioinformatics is now playing a key role in all healthcare industries, and especially the pharma industry, which is now, I guess, considering data science and bioinformatics a very important part of their portfolio. So I have a working experience. So if you can see in this survey, you'll find out that AI and big data has the biggest impact, right, in healthcare and pharma. So, however, to understand this application and to be able to find information from this big data, it's important to learn about the analysis strategy and get experience working on real data sets. Together with our research collaborators from top universities, we actually we have created and prepared projects for you that takes you through a project-based approach, where we take data sets from high impact publication and use them for practical exercises. It is difficult to imagine what the biomedical industry will look like in a few years' time. Sorry about that. So yeah, as I was telling you that it's with all this data coming from different sources and people are publishing every day, it's difficult to imagine exactly how this industry will shape up. But it is certain that there is a surge of biological data that is flowing all this time. This increase in data will form large innovative research projects. So one of the research projects that we wanted to talk to you about, that I wanted to tell you about, is the International Human Cell Atlas Initiative 10, that aims to create comprehensive reference map to all the human cells, and also devices, apps, wearables, and implantable technologies. So in our data science program, we will learn about these analysis strategies and methods, and we will learn about hands-on experience working on real data sets. For example, this is a project that describes modeling cancer precision medicine. So the application in cancer, the application in infectious diseases, we have projects spanning from agriculture, biotechnology, agri-biotechnology, and neurosciences. So wherever the application is, so there are plenty of resources from where you can learn in this program that we have prepared for you. So with this information, let me pass it on to Ms. Deepsha Biswas to talk about important deadlines and program registration options. Thank you, everyone. And it's to you, Deepsha.
Thank you so much, Dr. Mohit. Yeah, I will be showing everyone the different ways you can access this program. So the data science program is going to be starting on 12th of October. It is a 1-month program, and every day the sessions will start at 9 a.m. CST, which is the same time where this session also started. We will combine online sessions, self-guided study materials, and practical assignments for an immersive experience that has proven to be effective in our well-known and respected OmicsLogic programs. The program theoretical materials are found on EduTBioInfo. And I will just give you an overview of how you can access these different resources on the platform once you have the registration completed. So this is the link to the platform from where you can register to the different courses, and I will be posting the different links in the chat here. So if you go to this page, you can click on Get Started or Pricing to finish your registration. And we have 3 different options that you can choose from. We have the beginner option that covers all of the different resources that are going to be available for the program. We have an intermediate option that offers you mentor support and one-on-one mentor support in addition to the basic resources. And we have advanced membership for those of you who are interested in working on a long-term project with us, because this gives you access to more than one membership option, which means that you can go beyond 1-month program and get an additional 30 days more to work on your project and also interact with our intern. And you can also request for scholarships and discounts which are available. Please email us at marketing@pine.bio. I'll be putting my email in the chat, marketing@pine.bio, for any kind of request regarding scholarships and discounts. And once you finish your registration on this page, you see there is a coupon code here, which I will be sharing later on. And you can request for those codes as well. Then you will have access to what we call the EduT Bioinfo. So the EduT Bioinfo is the main platform for where you can access the courses. And this is the forum in the EduT Bioinfo platform. So from this forum, you see that we have a few topics posted here. This is like a classroom. Every participant of the OmicsLogic Data Science program will have access to this forum, from where they can use the forum to interact with us and with each other, access the different courses. So if you go under courses, you see these courses, and you can see the different courses that are part of the program, and also courses which are more relevant, like biomedical data visualization in R. So you have access to these courses as part of the program, and also projects. And there are some of these scholarship options that you can look at. So please visit this link if you want to apply for scholarship and let us know. You will also get access to what we call the Server T Bioinfo platform and the Code OmicsLogic platform. These two platforms are also a part of this program and will be freely available for you to use during the entire length of this 1 month data science program. And to introduce this program, I want to invite Ilya again to give you all a short demo. Ilya, over to you. Thank you so much, everyone.
Thank you, Victor. So as Victor mentioned, the program is intended to allow you to develop skills that cover a broad application of data science, starting from processing of different types of omics data and all the way to visualization, machine learning, and statistical analysis. All of these are equally important as you think about applications in cancer biology, as well as other types of biomedical challenges. So I'll briefly introduce 3 resources. The first one is going to be the server platform. This platform allows us to analyze different types of omics data using a guided and visual interface that gives access to anyone without any technical background to complicated tools and different types of visualization. So let me first of all explain how that works. Inside each one of the sections, you have an interface that packages these public domain published algorithms and provides a guided flow of data with color-coded maps, explained methods that work with a variety of input and output formats, and help you create reproducible workflows. As a result, you get to interact with the data without having the computational infrastructure needed to process these huge data sets. So let me briefly give you an overview of what that really looks like. So this is the link that you can follow to find this platform. Here you can see different types of data and different types of integration, modeling, and analysis tools. So I'm going to just briefly show you one of the demos. Each time that you select an option, there will be an explanation that guides you through the logic of analysis, an important component that is oftentimes overlooked on other solutions because they expect the user to know all of the inputs and outputs of each method and be able to navigate the process of analysis from an expert point of view. So this really eliminates that expertise requirement and allows you to use the platform and get results that are structured and ready for analysis. So this platform will be useful for processing big data, accessing tools that are difficult to install and difficult to run in a command line, but it will give everybody a quick sense of how to apply these types of methods to large next generation sequencing data sets. and we'll see different types of outputs and factors required to understand how to build such bioinformatics pipelines. The second resource that I would like to mention and give you a little overview is what we call Code Playground. So this Code Playground is intended to get you started with coding in both R and Python, interacting with the prepared data sets. After they have been processed, you can actually start loading them, visualizing them, doing statistical analysis, and an introduction to machine learning. Obviously, processing omics data is going to be very important in a variety of life sciences and biology research, not only to biomedical data sets, but we will primarily use examples with biomedical significance because we believe that data science is very impactful in that area. So let me show you how the Code OmicsLogic platform actually works. In many of these pipelines, one of the outputs that you will receive is a package, a code with the input data that you will later on can use to analyze in R and in Python. So this is the link, as Beats already placed in the chat. This is the link to the OmicsLogic Code Playground. And the OmicsLogic Playground is actually free to register. So any one of you can go on this link and create an account for free. Once you are in this, once you are in the platform and registered, you can start with very basics. For those of you that have minimal background in R and Python, we go from the very basics. So for example, you can see here loading data, some basic visualization, reading DNA sequences, DNA replication, reverse complements in R, and other topics as well. So you can really go through the basics. The ones that have a star are premium courses, so you'll get access to those as a part of this program, or you can actually buy a very affordable subscription for just $10 a month and explore some of these different types of courses. Now, in the context of this particular program, we are going to be using a little bit more advanced type of analyses. So I'll just give you a brief overview. Here's an example of what's called principal component analysis. So before you start going into the coding, you really get to understand where this fits in the overall data analysis process, and you get to understand the data set that was generated on the T-Bioinfo platform that we just looked at. So here you can see the data set. It's a gene expression matrix with the names of samples in your header and the names of genes here in your row names. And what principal component analysis seeks to provide you with is an ability to simplify the number of features, which are thousands, to a small combination of vectors in this multidimensional space, and using the projection onto this space, you can understand differences between samples or objects in your data set and study the relationship between groups of different samples. So as you can see here, you get to learn the code. This is a code in R. So we load some libraries and then input, we specify the input file, load this file and convert it to a matrix, and analyze this matrix with a principal component analysis library from the autoplot library. So then you actually get to try this code yourself. So here, this code could be run right inside the browser. You don't need to install anything on your computer. So you can see here that we have a data set, and we can just press on run. And once you press on run, it launches a cloud-based script that performs whatever is right here in your code block. So you can see that we already finished, and there is a an output here in the console, as well as some visual outputs. And so based on these visual outputs, you can see that some samples are far away from the others. And here you can see how the different types of expression data is distributed. And also you can understand the significance of the different principal components. So obviously this is just a starting point to start understanding how the data could be visualized based on all of the available data. And eventually we get to this kind of an output, which shows us that actually these are breast cancer subtypes and they are different by groups. So we see a group of luminal samples, a group of basal samples, and here we have a group of normal-like and cloud and low. As you continue through these courses, they will go from very simple, like how to load data, to interactive, like this, what we did, to eventually challenges where you have to write your own code from scratch and analyze data sets that we discuss in the program. All of this information is going to be both in R and in Python. And we do this so that you can actually have a sense of how these two languages work and develop some skills by practicing these different basic types of visualization and more advanced types of data science methods that are going to be important for any area of research as you start dealing with this kind of data. So when we go through this code breakdown, we really understand what the code does and can practice what it should provide us with before you start applying this in your own environment, your own developer environment. So we also go through an introduction of how to set that up for Python or for R, what types of libraries to include, what kinds of methods you should have. And as a result, for many of you, not only will you understand the syntax and read the code to understand what is being done to the different data sets, but also you'll be able to develop a library of snippets of code that are going to be very useful if you are a student or if you are conducting research and regularly have the need for different types of analyses that we describe and we use in this program. So again, I wanted to remind everyone that the program itself will start on October 12th. This is coming up very soon, so we have a short window of opportunity to complete your registration. As Bipsa mentioned already, we have a number of research scholarships that are available for those of you that are interested. Bipsa has also provided a link, and maybe she'll put it again in the chat for those of you that want to participate in the program but really require an assistant, financial assistance. We have several of those available. But let me briefly describe what we will cover in the program itself. So the first part of the program in week 1, we are going to speak about different types of omics data. We'll discuss big data challenges and opportunities, which many of those challenges have resulted in these pipelines to process data. Some of them have an impact on how we prepare the data for analysis, looking for batch effects and outliers. And some of them are important to understand the technologies producing these data sets so that we can incorporate their specificity to the downstream analysis. And that's why we'll talk about cleaning, loading, and processing these data sets and learning exploratory data analysis. So instead of jumping into a full analysis, it's always important to understand what kind of variation is present, what kind of information am I able to get out of this data set, and what technical issues I need to address before trying to run an analysis. And we'll have some hands-on sessions where we'll do practical use cases for different types of methodologies. Now in week 2, we will start talking about machine learning and artificial intelligence. We'll start from what's called data mining. So we'll discuss unsupervised machine learning methods like principal component analysis as an example, or clustering. We'll talk about how different methods use different assumptions about data and what do we need to understand about those assumptions to make sure that we are using those appropriately. Then we'll talk about supervised machine learning or classification. And when we talk about classification, we'll learn about methods like discriminant analysis, random forest and decision trees, as well as support vector machines and other types of machine learning, including deep learning. And we'll think about how those can be applied in the context of different project examples that we've described so far, including cancer, including microbiome, and including infectious diseases, so sequences of viral genomes. So we really will think about how these different methods are to be applied, and we'll also think about how to explore the models, so how accurate and how valid they are, both from a statistical perspective, but also from the perspective of biology and biomedical applications. Now, in the last session, we'll actually talk about how to get results with machine learning, because really it's not just about training a model and always looking at the prediction. There are many different use cases where different outputs from these machine learning models could be very useful to extract information from the data that we have. And so it's very important as you learn about these different types of methods, different types of applications of data science, to then consider a project of your own. So the program is designed to last for about a month, and we have numerous participants that have been able to use this time to both learn and develop an independent research project that will demonstrate how they can apply these different types of methods to a data set that is interesting to them. So we'll have a number of sessions where we will describe different projects, read publications, and explore publicly available data sets that you can start leveraging for your research. Our previous participants have been able to understand bioinformatics for the first time and learn how to apply data science methods to their own research data sets. So for example, Olga Udartseva, who is a research affiliate, a postdoctoral student at Roswell Park Comprehensive Cancer Center in New York, was able to benefit from this program, completing a variety of courses and really understanding how these different courses show the application of bioinformatics to preclinical data sets, and eventually how they can lead to insights about treating cancer patients and diagnosing different cancer patients. Now, we were also fortunate to work with a variety of participants. Bipsa shared a link here to different project examples that you can explore, how different people were able to use the program to develop this independent research. And also we've gotten some important insights from our participants about how our programs are different from your standard courses on Coursera, edX, and other types of online study platforms. So we were really focused on this research aspect and really making sure that our materials are very practical and very applicable to independent research projects. So this is a little overview of the program that we are going to start on October 12th. Again, the program is going to be called OmicsLogic Data Science, and it is designed to introduce you to the analysis of big data, to explain the role of bioinformatics in life sciences, and to help you wrangle large data sets and extract meaningful information so that you can do independent research in life sciences using data science skills. At this point, I would like to open this discussion for any questions. If any of you have any specific questions about the program itself or about any of the topics presented by Dr. Mohit Mazumdar or Dr. Harpreet Kaur about their experience and about the way that they will leverage the examples from their research, in the context of the training that is provided in this data science program.
Ilya, there is a question from Shreya. So she did ask that I'm in my first year of MBBS and inclined towards bioinformatics. I miss studying mathematics. What should I do?
Well, I'm not sure, what is MPBS again?
MBBS is medicals.
Oh, yeah. So we have some experience dealing with medical students. We work with Georgetown University Medical Center and their program for systems medicine. And a lot of the participants of that program go on to become members of a medical school. So what we've seen is that there is a tremendous growth in data-driven insights for diagnostics, for treatment selection, for even understanding different types of costs associated with treatments. And so it's very important for medical students to learn about how data is going to be used in the future of medicine, really driving the future of medicine to become more personalized, as mentioned by Mohit Mazumdar, and more precise, as mentioned by Dr. Harpreet Kaur, when she described the biomarkers. So we really believe that this is an important component of your clinical future. And if you are going to be exploring careers as a clinician, as a technician in a clinical lab, as a decision maker from a health policy and ethics standpoint, I think these are very important topics that are being discussed actively in the context of medicine. What role does data and different types of omics technologies and different types of analysis methods? NIH has, the National Institute of Health has a major push to enable clinicians to leverage data science and to also understand the implications of these types of methods like machine learning and AI. And so I think it's very important to consider both the ethical and the clinical aspects of how these methods work. Okay, any other questions?
Harshita, we will be emailing you regarding that. We have a program coming up on NGS, and also we have dedicated programs on RNA-seq and metagenomics starting in November and December. So I will email you those details. Does anyone have any other questions to add? Or you can also mention if you have any specific area. Ilya, if you could address Shreya's question, would it be too late to start learning bioinformatics and data science after completing MBBS?
Well, I think that the key here is to really be able to understand that bioinformatics could be application of bioinformatics and data science, or could be a degree to make you a researcher in bioinformatics and data science. And these are two very different kind of paths moving forward. I think as a clinician, as someone that wants to do research with clinical data, with patients with biomedical topics, you do not have to be a bioinformatician or a data scientist. But if you want to be a good clinician and a good researcher for biomedical projects, you do have to have some mastery of data science and some understanding of bioinformatics, because you will be required to deal with data and make decisions based on data. So how else will you be effective at that if you don't understand data science and bioinformatics? Now, the good thing is that you can do this in an effective way in parallel, I believe, with your MBBS. So you could really study short-term courses on data science, like the course that we are describing today. You can have a level of mastery that allows you to start dealing with data sets. And as these types of data sets come up in your program, you will understand where else you need to improve your skills and where else you need to develop a deeper understanding of these types of analysis methods, potentially preparing you for a future that is more bioinformatics focused. And I've interacted and met with a number of researchers as well as industry professionals and clinicians that have MD PhDs or that work in industry like pharmaceutical companies, biotechnology companies, different clinical research centers, and even just clinicians in the areas of oncology and infectious diseases that regularly rely on bioinformatics to make decisions and to conduct research. Even though they do not have a formal degree in bioinformatics or data science. Now, did that answer your question, Shreya?
You're welcome, Shreya, and thank you, Ilya, for answering the question. If we do not have any more questions, then we would like to conclude today's session. You can again reach out to us at any time. marketing@pine.bio is the email ID. Please use it and reach out to us. We will be emailing anybody who has left their email addresses with us regarding all the details of the upcoming programs. Thank you so much for joining today. We'll see you at the session. Thank you.